So this is my great pleasure to introduce Mathieu, Joron, working at Montpellier, long time. Is Montpellier? Yes. No. Yes. Yeah. It's just I have a dyslexia with Montpellier and Marseille. I don't know why. So I always mistake. So he's been working with butterflies and supergenes in butterflies for very, very, very long. He's a CNRS researcher, also for a long time, and so it's a pleasure. So he will finish this session. Thanks. And normally...
Thank you, Ricardo, for introducing me and pointing out how old I am. It's mostly the work done by Paul when he was doing a PhD in my group, and also other people who contributed to the work. So inversions, I'm not going to teach you too much about inversions, but they are important as genomic architectures which suppress recombination, and they are often involved in adaptation, speciation, or polymorphisms, and these are the cases where this recombination suppression effect is thought to have an important effect. Supergenes are a special architecture, usually involving inversions, and the definition in the literature is multiple tightly linked loci controlling distinct phenotypic elements, this is an important point, which segregate together in populations as a single and polymorphic Mendelian locus. The example I put is this rough example with multiple behavioral and morphological and also physiological traits which segregate together with this inversion. And the three morphs, so there are three morphs which coexist in the populations. So balanced supergenes are, this is my diversity of supergene slide. So they are repeatedly now found in very diverse organisms and involved in very diverse processes, like ecological processes, but they are very often, most often, they involve behavioral and morphological and sometimes physiological traits segregating together. And we find that most of them are seem to involve some structural variation, very often inversions, sometimes big deletions, and some of them are still to be discovered, probably. So supergenes are supposed to reflect selection on alternative complex forms, and this is where the recombination suppression is supposed to be beneficial. The term that is often used is coadaptation among different alleles. On this diagram showing a fitness landscape, you can imagine the different forms or the different alleles of a supergene corresponding to different fitness peaks, and recombinants being lower fitness and therefore falling in fitness valleys. So, but how can this evolve? The first problem is that inversions are not born differentiated because they come from a putatively recombining population. So there is no specifically an ecological advantage associated with an inversion unless it affects directly the genes or captures a series of genes which by chance happen to be co-adapted. But those genes need to be somehow segregating before the inversion comes. And so this is the problem. When rare, inversions differentiate via mutations only and not recombination, and this is very difficult to build and adaptively build a co-adapted haplotype if you only have extremely rare recombination. So how can we form inversions which differentiate and contain co-adapted mutations, which seem to be necessary to cause the initial rise in frequency of these inversions? So this is a kind of a conundrum that is sort of part of the interest of most people here, probably. So one mechanism is peak shift, and this is, basically this is the process that is supposed to be slow, and because it requires accumulating beneficial mutations on the same haplotype repeatedly. You can imagine also an inversion happening and diverging in two separate populations where they are fixed, and so where they recombine locally, and then come back into contact through introgression. And of course, this is what we found in our butterflies. And you can also imagine an inversion being a macro mutation and capturing at the same time a beneficial allele that doesn't need a coadaptation, and a breakpoint effect, for example. And in this case, you can have an immediate advantage to an inversion without having to have multiple mutations on the same haplotype. So the key questions are, why do inversions increase in frequency? And so do they preserve co-adapted alleles? And how do they differentiate? And why do they not go to fixation in the case of supergenes? So we work on Heliconeus numata, this butterfly with a very nice color polymorphism, living in the Amazon basin. It's toxic, and it's mimicking other species almost to perfection. So each of the forms is a precise mimic of another kind of a series of species. And you can see that it's very important in those cases for each of the forms to have the precise arrangement of different color pattern elements. And these are the different elements that we think are co-adapted and maintained together via this supergene architecture. So this is why different wing pattern elements segregating together. So we know it's found somewhere on chromosome 15, and we've discovered a long time ago that it was maintained by, associated with chromosomal polymorphism with 3 inversions, making 3 different chromosome types, which I call supergene alleles. So HN0 is the standard, non-inverted. HN1 has one medium-sized inversion, 400 kb, and is associated with this dominant form, which is very melanic. And then there's a series of 2 more inversions linked to the first one, so making a very complex pattern of inversions, and associated with another series of intermediately dominant forms. And the entire thing contains about 75 genes. And so there is a nice, you can see the progression. So there is we know which one is most derived and which one is less derived. And they're relatively old. They're about, so the first inversion is about 3.4 million years old, and the other ones are a bit younger. So how did they differentiate? As there's a lot of differentiation, and it's been kept for about several millions of generations. There's also an interesting pattern, a geographic pattern in this inversion polymorphism. So the 3 types are only found near there's no pointer, near the Andes. This is where the 3 forms are found, and then if you go into the Amazon basin, the lowlands, you only find 2 of the forms, and so you don't find the first inversion. So the sort of intermediate step is not found in the Amazonian populations. And then if you go to the Atlantic populations of Brazil, you only find the standard arrangement, no inversions basically, and little polymorphism. So there seems to be some, and we know that the first inversion came through introgression from another species 2 million years ago, and so we think that the polymorphism originated near the Andes, and then somehow some inversion forms invaded the populations in the rest of the range, but not the first inversion, only the haplotype with the extra 2 inversions. Predation measures showed that indeed those inverted haplotypes have an advantage in terms of mimicry and ecological and survival as adults. I'm not going to talk too much about that. But so there is better. The inversions bring better protection, probably through coadaptation. And so we wanted to see if there was signs of multiple loci being causing the wing patterns and trapped in the inversions. So Manon and Paul did this association study on this wing pattern diversity through the range, and then they only used the 3 inversions, the chromosome with 3 inversions. which has diversity of forms and which occasionally recombine because they make homozygotes. I'm not going to talk too much about the methods, but basically we have a tool to analyze wing patterns quantitatively through morphometrics and modeling, and we can also partition the wing into different elements or different wing regions, and so we can focus the association study on different types of variation. So the phenotypic variation can be looked at on a PCA, basically. This is aligning all the wings onto each other and plotting, making a PCA on the variable pixels. So it makes a very nice diversity, which you can then associate with genetic variation on resequenced genomes throughout the range. And to make a long story short, the wing pattern diversity is associated with specific loci within these inversions and only within the supergene, so nowhere else in the genome. Within the supergenes, we find specific and separate peaks of association on different loci within the inversions. And if you look at different, so if you look, for example, here on the anterior or posterior wings, you find sometimes the same loci being associated and sometimes different ones. So there is evidence that those different elements of wing patterns are associated with distinct genes or distinct gene regions in this locus. Some of them are well known, like cortex, now well-known wing patterning supergene, even though the molecular mechanisms are not very well understood. But there are other genes which we don't know their involvement in wing patterns, so they could be interesting candidates to look further. So yes, there is evidence that there is a coadaptation between these loci within the supergene, and so we found 14 associated regions. So this is an association study. There could be other effects increasing, perhaps artificially, the number of regions we find. So maybe some of them might be, so it might decrease, but we still have association with very distinct gene regions within the supergene. So yes, they freeze good combinations of alleles. So how do they differentiate? I'm not going to dwell on that. So we've seen a few years ago that this inversion polymorphism was initiated through the introgression of an alien inversion coming from a species where the inversion is fixed, and then introgressed in this and initiating the polymorphism. There was the introgression occurred about 1 million years after the speciation between those two species. So there was already at least 1 million years of evolution, independent evolution of the two inverted and standard regions, allowing probably to build up some differentiation already. And then there's been 2 million years of evolution since then, with probably accumulation of mutations. And so why did it not go to fixation? So there's 2 million years of evolution of maintenance of polymorphism, which is 8 million years of 8 million generations, perhaps. So there's something maintaining that and capping the frequency of the inversions, because so the first inversion, so one of the haplotypes, or the inverted haplotypes are very much favored, at least in the Peruvian populations. So we should expect them to go to fixation, but they don't. So we showed a few years ago that there was dissociative mating, maintaining the polymorphism. So these morphs actually prefer to mate with a different morph, which is very special type of mate choice or mate preferences. And theoretical work done by Ludovic Maisonneuve and Violaine Llorens showed that actually this type of mate preferences evolve readily when you have heterozygous advantage. So we thought maybe this is reflecting heterozygous advantage. And so we looked for possible problems with those inversions, and especially accumulation of deleterious mutations. So Paul looked at the very, very crude indication of accumulation of something is that the inverted segments tend to be much larger, like about 15% larger than the standard ones. Indicating that they have inflated with time and probably accumulated genetic material but we still have very good synteny, so it's not genes. It's actually TE insertions. So the insertions are full of TEs. So this is what you see on the right, compared to the segments in general. So what's really inserted in those segments, the inflation is caused by the insertion of TEs. I'm not going to be too long on the other types of indications that there are deleterious mutations. But Paul also looked at the ratio of non-synonymous and synonymous mutations and other measures that we do to indicate that there is a negative selection acting on these mutations within the inverted segments compared to the standard segments. So basically, to cut a long story short, inverted segments tend to have higher levels of non-synonymous polymorphisms and non-synonymous mutations generally than ancestral segments. And you can see the pattern is repeated for the three inversions in color here, with the standard one on the upper left and the inverted one in the lower right. So there is an enrichment of, there is basically a mutation and load associated with those inversions. So they have captured and accumulated deleterious mutations through probably bottlenecks associated with the formation of inversions and also the introgression, and lack of purge because of repeated or a sustained heterozygosity. So why are they sustained heterozygotes? We looked for a manifestation of the genetic load. And so Mathieu looked at larval survival in those different genotypes through crosses. And so if you look at the larval survival shown here on the y-axis, homozygotes for the inversions have a much lower larval survival and therefore don't go to adult mostly, when the heterozygotes have a good survival and roughly the same as the standard homozygotes. So this indicates that inversions are indeed causing deleterious effects, especially the first inversion when it's on its own. So the haplotype with only one inversion seems to have almost zero or very, very small survival in the larval stage. A little bit better in the for the haplotype that has three inversions, including the first one, which is something that puzzles us, because this second, well, the most derived chromosome type is in fact homozygous for the first inversion, but it seems to have recovered some survival in the larval stage. And all the heterozygotes are good, including the HN1, HN123 heterozygotes, which are in fact homozygous for the first inversion. So the second and third inversion seems to restore somehow or be associated with a lower load in the HN1 in the first inversion. Something we want to pursue perhaps. So heterozygote advantage, yes. So there's, we don't know whether this heterozygous advantage is actually directly caused by the deleterious mutations that we have found in these inversions. They could also be caused by breakpoint effects, which we don't measure with accumulation of deleterious mutations, and also perhaps possibly with some incompatibility with an alien inversion coming into a host genome. When this inversion comes homozygous, it might be causing some trouble. Those are hypotheses which we'd like to investigate further. So yes, they don't invade completely because of unpurged deleterious variation and also because of negative frequency dependence due to heterozygous advantage. So this is to highlight the dual roles of recombination suppression. It preserves beneficial mutations. So it's very important for, the inversion participates in the build-up or in the formation of very good ecological alleles, and which explains the initial success of those supergene alleles. But then it also captures a genetic load expressed in inversion homozygotes, which favors heterozygotes, and therefore it actually favors ancestral or standard alleles in return. So there is the dual role of recombination suppression is positive, negative. It's frequency dependent and directional in our case. I wanted to highlight as a conclusion that this effect seems to be, to us, inherent to inversion formation. So it's basically there's no way to escape the recombination suppression, and therefore it can have both roles. It's found probably in many of the cases that where supergenes have been associated with possible homozygous deleterious effects. As a conclusion, we could say that, so we think that recombination suppression evolves in response to a selection on the polymorphism and selection on the coadaptation of those alleles. But we seem to find actually rather the other way around. In fact, the polymorphism evolves because of recombination suppression. And so the recombination suppression is instrumental in building good ecological haplotypes, but it's not per se the coadaptation that is, it's not the duality between the two alternative ecological alleles that is causing this. So did the supergene form because of selection alternative mimicry optima or selection optima? No, because it formed because of the dual effects of recombination suppression. And then the second conclusion is that the paradigm of adaptive coadapted haplotypes may be a little bit misleading because the negative frequency dependence is not due to wing pattern polymorphism. And that's the same question. I also wanted to highlight as a sort of a follow-up that this is also to follow up on yesterday's talks, that we also are scanning the genome for more supergenes and maybe finding new ones. And this is a sliding window PCA that is done by Quentin and Benjamin currently in our group. And we're finding new inversion polymorphisms with alternative, or inversions, we're not sure they're inversions, but at least loci of low recombination showing alternative haplotypes, which you can see on the sliding window PCAs, like here, and this region here of showing a kind of weird pattern is a kind of a signal that something is happening, and then you look more closely into there, and you find that indeed the PCAs show very good, nice separation of genotype groups. Here we found basically two adjacent, well, there is one inversion or one, like three genotypes, homozygous, and heterozygous. And then the same thing here on an adjacent position. So we have two putative inversions in some LD, but not complete. So we're combining with each other. And with also patterns of strong differentiation, like probably not as old as the supergene, but still requiring an explanation in terms of maintenance of polymorphism. So that's for the future. And then I acknowledge the people who did the work, the people who helped with the sequencing, other people who contribute in many ways, and also the funding from the French ANR and the ERC. Okay, thank you.